This is Audiomunity, a podcast about our body's never-ending fight with the outside world.
Hi, everyone. Welcome to Audiomunity. This is episode 57. We are here recording on Thursday, July 2nd, joined as always by Kevin Bonham.
Hello.
And I am Matt Woodruff. And Kevin, people might have heard the end of the last episode where we suggested that if I was in a different office, it might be something significant. And I will say that the reason for the office shift today is actually not the potentially dire outcome that we discussed the other day.
Yet.
Yet. Yeah. Yet. But no, we have childcare summer break this week.
It's a good time.
Yeah. Yep. So
mine was a couple weeks ago. Thank you for jumping on here. We're unprecedented recording 2 weeks in a row to make up for the 2 months that we were silent.
That's right.
And in the last episode, we did all politics all the time, and this time we're taking a hard pivot into pigeon navigation.
Yep.
Which I think is perfectly apt in terms of discovery science, in terms of why expert peer review is maybe important. So this is going to be fun. I think.
This is a good rebound paper for us. Good rebound paper. Mm-hmm. My dog is barking outside. I'll be right back.
Okay. Yeah.
So it is 100 million degrees here in Atlanta. Heat index hit 110 earlier, and I have a Bernese Mountain Dog at home.
We're going there this week here as well.
And
I've been
can't leave the dog outside
to camp. I've been biking him to camp and just like drenched in sweat when I get to the tee.
Yeah,
super fun.
Yep. So I mean, we don't want to hit politics hard today. But I will say that if you have interest, we talked a little bit about some proposed OMB rules in the last episode. We spent a lot of the time, actually, probably too much time at the beginning, talking about the grant review process again. But one thing that we really jumped into pretty hard was the need to have scientific review done by scientists that know what they're talking about and know something about the field. And so here's my caveat for today's paper. I don't know anything about magnetism. It's been a long time. It's been a long time since I learned about magnets. I don't think I ever truly knew deep down how magnets worked.
No, I definitely took physics my first year at college, and that was the last time. Are you familiar? You know the I think it was Insane Clown Posse or something. One of these musical groups. So there's some meme on the internet that's fucking magnets. How do they work? As a nobody knows anything.
Yeah.
Some people do know how magnets work.
It seems like it. Based on this paper.
We're not those people.
Yeah, based on this paper, it does seem like at least a handful of scientists out there know how magnets worked. And I will one-up you. I dropped out of college physics because I waited until the very end, and then I argued to them that it was going to interfere with my ability to go interview for PhDs. And they were happy to let me to drop out on it.
Congratulations.
So, yep.
I took there were two tracks of physics you could take at UCSD. One was the engineering and high level calculus infused math, and the other one was we know med schools need physics, and all the people that just want to get it out of the way. I took that one. I was never going to go to med school, but I definitely took the easy way out. So the paper that we have been alluding to all this time is called, it's published in Science.
It is.
And it's called Homing Pigeon Navigation Relies on Super Paramagnetic Macrophages Under Overcast Conditions. Sets out.
Yeah, this is a fun thing about science and nature, right? I had the privilege of being able to publish in Nature one time. And that was around COVID, and we were doing some cool things with B cells and autoreactivity and all of that stuff. The cool thing about being in Nature is like you're excited when you get your stuff published in there because you're like in the physical thing that gets distributed out to all the people, right? Which is really, really neat. And you do that and you get it, and there's a picture of a frog on the front.
Yeah.
Right? Like it has nothing to do with immunology at all. There was an article about the Mars rover in
Yep.
that article. That was actually one of the feature articles, and I think it's really cool that Science and Nature really stuck to these roots of like, you know what, we're going to publish stuff that we think is really cool, right? And this is one of those things like, how do pigeons work? Well,
Mhm.
actually, they work with magnets. So
Evidently.
there you go.
Maybe.
Maybe we'll get
So
there.
we should finish. So the
Yeah.
the authors here. So there are 3 corresponding authors, a sole first and 2 co-lasts. And so the first author is Clivia Lizowski, and the last 2 authors that contributed equally are Martin Wikelski and Christian Kurtz, and these folks are all in Germany. So Institute for Molecular Medicine and Experimental Immunology at the University of Bonn in Germany, and then Department of Migration at the Max Planck Institute of Animal Behavior. Cool.
Which is awesome.
Very cool.
Yep.
So by way of introduction, I just wanted to say basically everything that I know about this topic, I know from reading a book recently by the science author named Ed Yong. He just wrote a book that's called An Immense World, and it's all about all of the different ways that animals sense the world. So he has a chapter on vision and hearing, of course, but also stuff like heat sensing, and there are these beetles that apparently have these tiny little organs that expand minutely based on heat. They like trap a fluid inside, and so they expand a little bit because of heat, and so they can sense, like they can like go towards fires.
Cool.
Because that's how they breed or something. I don't know.
Right.
He has a section on like electroreception, and the section on magnetoreception, which is what this paper is about. One of the things he talks about is that this has been postulated for a very long time, and people, at least at the time the book was written, and we'll argue about whether we know anything more now. Basically, no one could find the magnetic sense organ, which makes it very hard to study if you don't know where in the body magnetoreception is located. But I think there is good evidence that animals have, and pigeons included, have some ability to sense the Earth's magnetic field. Other animals, they kind of go through a nice little, it's a very short introduction, but they kind of talk through a few of the other animals and hypotheses that have been brought up. So like things like turtles are thought to be able to navigate based on the Earth's magnetic field. Recently, there's been a lot of work on bacteria that have these little organelles that contain, I think it's ferritin or something like that, and that allows them to orient in some way to magnetic fields. But as I said, for animals, for chordates especially, there's been no detected sense organ. People think it might be in the nose. I think I learned when I was a kid that it was in the nose.
Yeah.
There's some magnet in the nose that allowed them to point in the right direction. But there have been experiments where you like put birds in a completely blacked out room with no other senses available, and they can still orient in the direction of home. Which is pretty neat. At the end of this paper, figure 4 of this paper is like,
it's amazing.
Fantastic.
It's a wonderful experiment.
They just release pigeons and then they track where they go, and they across miles of navigation on a cloudy day versus a sunny day. It's like,
that was great.
Amazing.
That's great. Yeah. And so I will, like I said, get to the caveat that as we jump into these figures, there are some physics-based assessments of the magnetic properties of some of these organs right over my head. So we're gonna make we're gonna make a lot of caveats in that. I'm gonna go ahead and assume that a reviewer is capable of assessing whether something is truly magnetic or not. I
Mhm.
believe that they can do that. I believe that based on figure 1,
There's magnets in the pigeons.
the liver and the spleen lines seem to be different
Yeah.
in the
They have
way
different
that they're
slopes.
behaving on that graph. I would even suggest that it looks statistically different to me. But
Good review, Matt.
No, it's
Solid analysis. The lines look different.
No.
I will say I did do a little bit of digging into some of these words because I was like, what is super paramagnetic? It's in the title.
Yeah, it is.
What does that mean?
Yeah.
So I learned a few things, and I'm gonna again caveat heavily that this is based on a few minutes of Wikipedia and Googling. So my understanding is basically, so if you think about a magnet that you would like stick on your fridge or like those little magnets that you can buy, they have a dipole. So they have these ferromagnets have a particular orientation. The electrons in the atoms of that metal are shifted in a semi-permanent way, right? So they have a pull. So the electrons are all shoved to one side of the magnet. That makes that side negatively charged. And that makes the other side, because it sort of has more of the protons without the electrons, shoved to the other side. And that is a stable orientation, right? That's called hysteresis, that it stays in that orientation. And what it means is as you pass a magnetic field across a magnet, it will orient, it will try to orient in a particular direction in a stable way, right? There are other molecules, these are paramagnetic atoms, for example, that have electrical properties such that if you pass a magnetic field over them, they will sort of take on magnetic properties and they will shift in the direction of the magnetic field, but they don't retain that orientation. If you took that atom and you moved it to another location with a different magnetic field, it wouldn't hold on to that directionality the way that a ferromagnet will. And they don't sort of, in this part I don't really understand, but what it sounds like is that if you have paramagnetic particles, they don't sort of cooperate with the other molecules around them. In a magnet, in a physical magnet, the strength of that magnet is in part due to the fact that all of the atoms in that particle, or in that piece, that physical contiguous piece of metal, are cooperating with each other and sort of build on each other for that magnetic orientation. The super paramagnetic bit that we're talking about here is sort of quite small, but not atomic scale particles. So I can't remember the exact, it's like 10 to 50 microns or something like that. So it's like smaller than the size of a cell, a typical cell, but they're not individual atoms. And the key here is that those particles, they don't have the hysteresis. They can't hold on to the magnetic orientation. But when a magnetic field is passed over them, they orient in a way that sort of compounds because of all of the atoms in that particle. So if you have paramagnetic particles, when you do this test, which again, I don't totally understand, but they have different sort of magnetic forces and they're doing them at different energy levels.
Mm-hmm.
If you have paramagnetism, where all of the atoms sort of will orient but independently, you get a linear slope. You get a linear change over time. Whereas when you have the super paramagnetic property, you get this curve. So as they're sort of like holding on to a shape, you apply more and more of this force, and then eventually they all sort of orient together, and that's why you get this steep slope, and then it sort of levels off. So the more S-shaped your curve is, the more it's super paramagnetic as opposed to just paramagnetic.
Right. And
why that matters, I have zero idea, but that's the extent of my learning on super paramagnetism.
Well, and I would say just from your description, it feels like that would be it would opened itself up to be a temporary responsive element rather than a sort of permanently oriented element, which is
Exactly.
probably what you need as an animal, for example, is orienting in different directions, right? You need a temporary response that then can reset, based on a new animal orientation.
Yeah,
So,
and it makes sense if you have some kind of cellular property or, sensing structure, the sort of force of the magnetic field can drive either through like mechanoreception or some other kind of cellular mechanism that we could imagine receptors that would understand those forces. And actually in bacteria, I think, and again, very heavy caveats, grain of salt, all that, but I think that the organelles in bacteria that have been discovered, um, those have hysteresis. They have this memory. So they're sort of like have a fixed orientation to magnetic fields. Whereas these ones that they're talking about, it's more about sensing changes. So I'm guessing like as a pigeon moves around, it's sort of these things are shifting dynamically and somehow that is sensed, or that's the argument that's being made. So
cool. So I'm certain that we butchered that.
of course,
But
yeah,
thank
absolutely.
you for at least trying. I was just going to hand wave
No worries.
away A through C. We get a little bit more into the immunology space once we hit D. In that people in the immunology now use histology, right? And so we're into a histology space, and the question here is actually quite simple, right? You just mentioned we understand that animals, organisms, probably are responsive to these magnetic fields. It is hard to square that if you don't have any sense of where in the body that might happen, right? Like if you can't find a magnetically responsive organ, hard to understand those things coming together. And so this is literally just a tissue sampling experiment where they took pigeons. They took the liver from pigeons. They took the spleen from pigeons. It's not clear to me why they took the liver and the spleen.
Well, so
But
they said their previous work. So they previously reported. This is the last paragraph. This is actually kind of cool. Their
I must
last
have missed
paragraphs
this.
of the intro.
Yeah.
We previously reported that splenic red pulp macrophages in mice and humans are intrinsically superparamagnetic.
Yeah.
This is due to their function of degrading damaged or aged erythrocytes, so red blood cells, which results in the liberation of iron from hemoglobin. So if you're a macrophage in the spleen and in the liver, one of your functions is degrading a bunch of red blood cells because that's where they sort of like aggregate, right? So you're soaking up all these red blood cells. They've got tons of hemoglobin. So now you've got all this iron. And apparently they're storing these liberated iron from red blood cells as in ferritin. And they say, so it's in a protein complex that sequesters iron in non-toxic form. These spherical protein nanocages can store up to 4,500 iron atoms in a mineralized form and exist as soluble cytoplasmic proteins are associated with vesicular compartments, such as lysosomes. So that's their previous work.
Mm-hmm.
Leading into this, they found these macrophages in mice and humans, in livers and spleens. And I'm guessing they thought, okay, well, we've got this. They apparently understand magnetism. So they've got these super paramagnetic macrophages. Why does that matter in mice and humans? Well, who knows? But where else might magnetism function? Hey, maybe in pigeons.
Yeah, maybe in pigeons. I like the idea of counting numbers of atoms.
Yeah.
I feel like in every other sense, we talk about like the astronomical nature of atoms. I really like the idea of putting into a paragraph 4,500 iron atoms can be stored here. Yep, that for some reason is soothing to me. So, all right. So, okay, you've got these different tissues from a pigeon, right? Liver, spleen, brain, eye, beak, maybe places that you think might, for example, be magnetic if you were to just like a priori guess, right? You said that you thought that they were in the nose, for example, so you could point your nose the right way, right? Like that's the fun kind of medieval thinking of like it must be this way because it makes sense to me. It's like the horns. Remember the goat horns jumping off cliffs, and
Yeah.
they have the horns so that when they hit the ground, they can stick with the
Yeah.
horns first, and that protects the rest of the animal. So, okay. So, surprisingly to me, and unsurprisingly to them, they found, cells in the spleen with, with the iron deposits that they're looking for. But interestingly, in the liver, really is where they see the concentration. This is just
Yeah.
a general histology sort of stain, right? Looking for iron deposits.
Yeah, they say elsewhere what the actual stains are. There's some dyes that I wasn't familiar with, but it also sounded like in a different one that they actually can do immunohistochemistry with antibodies for Fe3 ions. Did you
Yeah.
know that that was a thing? I
I
didn't know
mean,
that
I've
was a
seen
thing.
that for like gold, for example. So you can do immunogold labeling, or
Right.
you can
Okay.
you can you can.
Well, but isn't that you
No,
attach a gold particle and then you see it on electron microscopy because it's
yeah.
dense?
So yes, you're right. But yes, the answer is yes. I have seen antibodies raised against
Individual ions like this?
yeah, individual
Okay.
ions. Yep.
Cool.
So, but yeah, cool. And so what we've got here is we've got some general overview of different organs. Clearly, we're seeing a lot of iron in the liver in a surprising way, which I guess is not so surprising, right? Like we think about the liver, and the whole polar bear thing, right? You can't eat polar bear
Mhm.
liver because it'll kill you because of iron toxicity, right? So this is not totally unknown, but we've got a lot of iron hanging out in the liver. We go into that same liver, and now we're doing the kind of immunohistochemistry that you were talking about, Kevin, where we're asking the question, where is that iron located? And if you're looking in the spleen and thinking, well, spleens break down red blood cells, livers do that too, right? So there's a lot of blood filtration that goes on in the liver. One could imagine a similar sort of thing happening. One might imagine that macrophages might do that kind of thing. And sure enough, if you look in Figure 1E, we've got a macrophage population that looks like it's nicely marbling the liver there. And as we go in and do that co-stain, as you mentioned, we certainly do see an overlap in the MHC2 expression and iron deposits. Would say here, MHC2 shows up in a lot of circumstances.
Yeah. Yeah.
It
Totally.
is definitely on macrophages. There's no doubt about it. You can find it on a lot of things too. Now, probably it seems to me like these are phagocytic cells. There's no doubt in my mind, right? And we'll get to that later. But not a definitive macrophage marker here, right?
Sure.
MHC2 is much more ambiguous than that. But if you take those cells and if you sort on apparently just MHC2 out of the liver, and you do iron metabolism related gene searches after transcriptomics, what you find is apparently a fair amount of iron processing genes, essentially ferritin
Yeah.
heavy chain, heme oxygenase 1, transferrin receptor, excuse me, right? Like all things involved in iron metabolism. Now, I'm sure that they found a lot of other things
Sure.
that they are not reporting on here, but certainly, some core components appear to be related to this this iron pathway. And so they asked the question, what are these cells? And it seems to me like what they did was they took their transcriptomics, they put that against known transcriptomic signatures of immune subtypes based on their MHC expression. And they basically did an overlay looking for macrophage markers, B cell markers, and dendritic cell markers. I assume that they only did those three things. Could be that they did T cells or whatever too. And essentially what they find is that their isolated liver cells are mostly completely unique.
Yep.
Right. So we've got 7,043 genes that are only in these MHC2 liver cells that they've isolated. We've got an overlap of, and I don't quite understand how to read this, because clearly there are a lot of B cell markers that are probably going to be there that are clearly not being reported here, right? 9 seems like a low number of unique B cell genes from whatever data set that they're using. But
Yeah.
regardless, they've got an overlap in the B cell compartment of 12 genes. They have 9 genes related to dendritic cell markers, and then 43 that are shared between macrophages and these MHC2 positive liver cells, with 43 unique macrophage markers. So half of the canonical macrophage markers are not expressed in their population, and realistically, a small percentage of the genes that they are seeing are showing up in their canonical macrophage space. Now,
I will say the database they're pulling from is the Cell by Gene from Chan Zuckerberg Initiative database, which is focused on humans, and these are pigeons. So a lot of differences. Later on, they talk about this other cell type that are the pigeon equivalent of neutrophils. And I was like, I didn't know that pigeons didn't have neutrophils. They have something else that are like neutrophils. Makes sense, I suppose, right? We split off from birds quite a long time ago.
Sure.
From that lineage. But
dinosaurs.
Yeah. But all of that is to say, these signatures are based on mammalian macrophages. So it's not so surprising to me that you're not going to have super good overlap. That said, I do agree with you that the fact that the way that they do these Venn diagrams, I think, is a little misleading, because you have 7,000 versus 50 roughly. This terms it's 2 orders of magnitude separated, and the areas of those Venn diagrams are pretty similar.
They're pretty similar.
It's a little bit misleading.
Yeah. And I'm not going to sit here and say throughout this paper, I'll allude to it a little bit. I will not sit here and say that these aren't macrophages. What I will say throughout this paper, I think, is we don't know necessarily what the biology of these cells are. And we've talked about this before, I think, in other areas, actually, because I think season 2 really of autoimmunity has been immunology in interesting spaces that we don't expect it, right? And so we've had situations where, yes, you can call the thing a thing, but where does the biology actually begin, right? Have we just binned it, and do we truly understand it? So I would encourage folks, as we go through this, to not necessarily make deep assumptions about, exactly what these cells are, if there are subsets of them that are special, right? Those kinds of things. So
and also, I think, the other season 2 theme, immunology where we don't expect it, yes, but also what are concepts anyway? My repeated push to get listeners to become Buddhists, right? What is a macrophage anyway? And I think it is definitely fair to say we don't know what the biology of these cells. I think that's very clear.
Sure.
We do something that is probably phagocytic, probably chewing on red blood cells.
Yep.
And maybe has adopted an extrasensory function.
Potentially.
We'll see.
Potentially. We'll see. We'll come back to that. They definitely do take up dextran. There's no doubt about that. So they take up dextran, which is sugar, basically, complex sugar, right? So we have that, and we will get to collagenate, which essentially confirms their phagocytic nature in just a second. So we know that we have cells. We know that they are magnetic. We know that they're nicely marbled throughout the liver, a little bit in the spleen. We know that they share some properties with a number of cell types, although mostly the shared genes are in the macrophage lineage, and that those cells behave sort of like macrophages in a dish. So what happens when you blow them up? And so, another nice thing that I like about Science and Nature papers is it's like, we saw this thing. Boom. Right? Like there's not a lot of
We're
walking
not futzing
along
around
here. There's
here.
not, yeah, exactly. So, I mean, how do you know if they're important? The answer is you get rid of them. Classic immunologic technique. We're just going to make a knockout. Knockout pigeons, maybe they exist. I don't know. Who knows? I'm sure you could CRISPR knockout pigeons. I would suggest.
Are you sure about that? Because, like,
I'll bet you could.
I mean, maybe, but like, how are you gonna manage that breeding program? Like, where, like, you could put it in the eggs? Like,
yeah.
I don't know how pigeon breeding works. I will say that in The Origin of Species, Darwin spends a huge amount of time, like in the 1800s, the two big like breeding things that he spends a lot of time on is mouse breeding and pigeon breeding, because that was like a hobby of like how many different traits can you get. And of course, we've made huge use of mice in immunology because they're also mammals. That makes sense. But yeah, I mean, it's possible that you could do it with pigeons.
They bred them for fun behavioral traits, right? Like when we think about breeding programs for the delight of human beings, we generally think about dogs, right? And we're looking at lots of different physical characteristics, shape, sizes. The interesting thing about the pigeon breeding programs where they were like looking for interesting flying patterns and like way these things behave. So you could create a pigeon that like constantly looped, or one that like went up and then like dove catastrophically close to the ground before pulling up at the end, right? So they were messing with pigeon brains through breeding, which is, I think, sort of an interesting moment in
dogs
human
too,
history.
right? Dogs have all kinds of behavioral.
Yeah, but we like pretend that that's not as prevalent as it actually is, because it has terrible implications. So, we, I suspect that you could put an adenovirus into a pigeon egg and get a pretty decent, CRISPR-based knockout. But anyway, there is, and we've talked about this before, an easier way to knock out macrophages, and also anything else that ingests anything, and that is called a collagenate-loaded liposome. Collagenate-loaded liposome is essentially a little ball. It's a particle that you inject. It's got lipids on the outside, and on the inside, it's got collagenate. And collagenate is toxic to the things that eat it. So what you can do is you can take an organism and you can either systemically or locally provide collagenate-loaded liposomes. And functionally, anything that picks these things up through phagocytosis then dies.
And the death is not. They don't go quietly into that good night.
They do not. No, you can actually use collagenate-loaded liposomes as an adjuvant in a vaccine because the mechanism of death is inflammatory in nature. So, keep that in mind. We'll come back to that later. But figure 2A, you have a control liver. You add collagenate into the pigeon. And sure enough, anything that was MHC2 positive before certainly isn't now.
Yeah.
Actually, I think they're looking for iron. I think they're looking
and
at the iron deposits. But
yeah, exactly.
But we know from before that those are the MHC2 positive cells. If you ask the question, how many iron-loaded hepatic macrophages are there left in the liver? The answer is less. If you
not as much less as I would have expected,
right?
But they're doing counts.
Yeah,
they're doing iron positive. If you look at the histology, my guess is if you asked like the amount of iron per cell as well, like it's just way lower. There's just no concentrations of iron anywhere.
Yeah, there doesn't seem to be, and this is why you always quantitate images. Right? Because you also always provide the best image you have for the effect size or the effect that you're looking for. But regardless, you definitely do have a significant decrease in iron-loaded macrophages. They're doing groups of 4 pigeons here. If you go and you try to use your same column to isolate these macrophages, and I think we might have actually skipped over the most fun part of
Oh, yeah.
figure one, which is at some point they're like, hey, these macrophages are loaded with magnets. And then they think to themselves, well, sometimes we use magnets to sort cells. We usually attach magnetic beads to antibodies, attach the antibodies to the cells, and that's a way that you can pull out the cell that you want. Here, the cells themselves are magnets, right? And going back to your background, they had already known that from their work in the spleen. And sure enough, in 1G, I think it is, you can, let's see, let me make sure of that before I just say words. Yep, 1G. You can actually just take suspension of cells from the liver and put it over a magnet and get
Yeah.
cells that come out,
Yeah,
which
this
is cool.
This is actually I had meant to talk about this before because I remember very distinctly my first job as a postback back this would have been I guess 2006, 2007. I think that this idea was fairly new, right? So we have our fluorescence activated cell sorters that everybody knows about, where you attach antibodies that have a fluorophore on them and you send them through a fancy machine that, based on whether a fluorophore is active, it'll send you down different tubes. This idea of MAX, magnetic activated cell sorting, I don't know what the A stands for there, but basically you have antibodies attached to magnets, and then you incubate those with cells, and then you put them through these columns with very strong magnets, so the antibody bound cells will hang out. The other ones that you can like pull off the supernatant while your cells are bound. So this idea has been around for a while. I was saying before we recorded, I wonder how much that original finding was because they were trying to isolate some other cells from the liver, and they kept getting macrophages, and they're like, what the hell is going on with this?
Yeah.
And it's because their magnetic sorting was just pulling these other cells out due to intrinsic properties.
Yeah, no,
That's my guess. But
Could well be. Could well be. So, in figure two, when they've injected coladronate into these animals, the question is, can you continue to isolate cells? No, you can't anymore. So, or at least visually, you can't, right? We know from 1A that there are some cells still there, but they're certainly not coming out of the magnet in force. We can look, I'm going to skip to D, whether we, can fact sort and find those same MHC2 positive side scatter high macrophages in the liver. The answer is coladronate, as it does quite effectively, does in fact deplete those things. And the quantitation of that is off to the right. Yeah, I mean, basically, you get rid of these things using coladronate, right? Like that's the take home, right? You inject coladronate loaded liposomes into these animals, and you look sometime later and you get rid of both the cellular and magnetic properties of the liver that you found in figure one. Fair summary?
yes.
Cool. Because then we want to know, but yeah, well, where really? Right? And so figure three is essentially a dive into the actual structure of the liver. And essentially, without getting into extraordinary detail, because I want to get to figure four as quickly as we can, because I think it's a lot of fun.
Mm-hmm.
Without getting into extraordinary detail, there is a lot of complexity in the liver filtration system, right? Blood goes through, it's being used as a heavy filter for essentially everything. And also there are very specialized, just like in the kidney, just like in the spleen, right? There are highly specialized cells that sort of line those different structures. And there is a significant nerve fiber engagement within this structure, right? And there are specific macrophage-like cells that seem to line these nerve fibers, right?
Yeah,
and so
this was a surprise to me. I mean,
Yeah.
not that I know a ton about liver anatomy, but I know at least a little bit about liver anatomy. As I said in one audio immunity a few weeks ago, it has something to do with digestion. There's like coming off of the large intestine, there's the portal vein that goes directly into the liver, and there's a bunch of stuff like it goes directly there to basically be detoxified before it enters widespread circulation. So like portal vein blood has a lot of different components, metabolites potentially. You go through, you filter through this sponge of the liver. Liver's an amazing organ for a lot of reasons. But yeah, I was a little bit surprised at how much enervation there is there. I do not think about that much, although I think increasingly it's clear that there's a lot of crosstalk between the nervous system and the digestive system that is not conscious, right? That it doesn't rise to the level like, I can't feel my liver. But clearly there is a lot of sensing happening that is below the level of conscious awareness through things like the vagus nerve. We know, for example, you can change inflammatory posture if you sever the vagus nerve in mice from some microbes. There's this whole field of what's called enteroception, like sensing of your internal state, especially like digestive properties. So I guess I shouldn't have been surprised, but I was kind of surprised, or at least I had never thought about this.
Yeah, and so what we're sort of I think alluding to here is almost this idea is the liver as a transit hub for those signals, right, from the direct, really complex microbial environment that is the gut into the brain where it can respond rather than it being a more direct route. So, yeah, I think we can leave it there. There are MHC2 positive. Right. We have cells that seem like the right cells, iron-packed cells that seem to be aligned with the nerves that are running deeply through the liver. If you take coladronate, you add it to the system, those cells seem to go away. Right. So those associations seem to be at least some of the associations that you are destroying when you add in coladronate. So then we get to the fun figure, which I think is why we picked this paper.
Yeah.
Figure
100%.
4. So now you have a mechanism to knock out macrophages in pigeons or everything that ingests a coladronate-loaded liposome. And you ask the question, well, if this is involved in homing, magnetic homing, I'll bet these pigeons are going to run around in circles and bump into each other. And the best way to do that is to let the pigeons fly all over the place. So they trained the pigeons to fly east to west, basically, right? So there was a training program involved in this, and then basically
this is a famously
Sure.
pigeons do this, right? Like we think
this
about
is a
pigeon
thing.
messengers,
Yeah.
and stuff. You take pigeons, you move them from one location to another, and they can home back to their original roost. This is something that people have been doing with pigeons for ages.
Right.
So,
yeah. Yep. Which is cool.
Known mechanism. And they can do it under many conditions, including overcast
they can,
and sunny
even
conditions.
when it's cloudy, usually. And in this case, what they've done is they've taken 34 total animals. They've injected coladronate-loaded liposomes into 27 of them, right?
No. It's an 18.
Coladronate liposome injection. Oh, no, because they're overlapping animals. Regardless, you're taking a subset of those birds, you're injecting them with coladronate, and you're asking the question, how do they do? Right? Can they still go back and forth? And so Figure 4b is an actual map of the tracks of these pigeons as they go back and forth east to west under various conditions, either under sunny conditions, overcast conditions, with or without coladronate. And C is something that I'm very used to seeing because that's generally how we track cells.
It's like a cell track.
It does. Exactly. It looks like cell tracks, right? But these are bird tracks over long distances. And I mean, the very clear takeaway here is that whatever you did to those birds, if you take the sun away, because the sun allows them to orient just fine, they do just fine, right? Which is interesting, right? Because it means they're capable of tracking movement in the sun in a dynamic way that allows them to stay on course. But if you put coladronate in and you get rid of the ability to see the sun, the birds simply go everywhere. There really is actually very little directionality here. They're clearly completely lost. Yeah.
Yeah, I mean it is stark. It's
Yeah.
like
No,
it's not subtle.
yeah.
This
No,
effect.
it's like in a cell you lost the receptor that allows you to respond to the chemotactic gradient, right? Like that's the level of problem that you've introduced to these birds. They clearly know they're supposed to go somewhere, right? Like they haven't just not moved, so they're flying, but they're not flying in the right directions. One or two of them do okay, but no, actually
I
one
mean,
maybe
by random chance, one of
by
them goes in the
random
right
chance.
direction.
One, yeah, probably you're right.
But this is they have indeed. I really love this. I don't know how they quantified it because they have a quantification: birds not homing,
Yeah,
which like to me it looks to me like all of the birds home in the control and in the sun condition, but and one of them homes. One of them goes in the right direction at least.
Mhm.
But it's 100% of birds not homing in the collagenate overcast, and like 20% in the sun and control conditions.
Yeah.
So,
Yeah.
but
So
like
I mean,
so clear.
yeah, very, very, very clear. I mean, C, D, E, and F are essentially all the same data, plotted in slightly different ways, which is something you do. Like, there's no criticism there. Just showing, the vectors, total vectors in E, and the track efficacy, which is obviously extraordinarily low. If you're going in random directions, you're not really getting closer to your endpoint. So, um, and that's where the paper ends, which is great. They're very, very clear effect. There's no reason to go on about it. When you add in collagenate loaded liposomes, you deplete a population of cells in the liver of these birds that stains with MHC2, shares some markers with macrophages, and when you add in a systemic macrophage ablator,
Ablation. Yeah.
those cells do go away, and these birds cannot home under cloudy conditions. So now let's get to the question, I think, at the end, which is, are we confident that these macrophages are the reason that these birds cannot home under these conditions?
So the way that I would say it is that they have identified a potential mechanism that, for all of the ways that they tested in this paper, you would expect for a magnetic sensing cell or organ. So they are very, very plausibly the way that pigeons use magnetism to navigate. I think so. If we look back again at the title, homing pigeon navigation relies on superparamagnetic macrophages under overcast conditions. I think that we could definitely quibble with macrophage as the way that these cells are labeled. But I think that, and macrophage is the reason that it was a hook for autoimmunity. So I think,
Yeah.
I'm happy that
and
they
to be
used
honest
that
with you,
phrase.
I actually believe that it is, I don't think that we've proven it. I think there's a lot of biology to investigate there. I think it makes a lot of sense to me that these would be a specialized subset of macrophages that could be responsible for this.
And
I think it's
from an
completely
evolutionary
plausible.
perspective also,
Yeah.
I think it is it feels very plausible that there is macrophages evolved to clean up red blood cells. One of the consequences of that is we build up these iron deposits in ferritin that have some properties that would allow pigeons to sense magnetic fields.
Yes.
Liver already has a bunch of nerves.
Mm-hmm.
There's already interoception happening.
Mm-hmm.
It seems very plausible to me that the sort of evolutionary step whereby you would go from a byproduct of homeostatic clearing of red blood cells in the liver to a sense organ that is relied on for navigation in conditions where visual signals are not like that. So that series of steps, there's a plausible evolutionary argument to it.
Mm-hmm.
The fact that you're finding these cells in the liver in pigeons, the fact that it's near nerves in the liver, Like all of those things make this seem very plausible to me. I agree with you that we don't know the specific cell, exactly what it is and what its biology is. We don't know the mechanism whereby whatever signals are being sent, like how it's being sensed in the liver and how that's translating to an ability to navigate. Like all of those things, I think, are open questions, certainly.
Well, I would go one step further than that because I think this could be happening. I think it is all very plausible. I think it's probable. Collagenate-loaded liposomes knock out everything. They knock out everything. They knock out dendritic
By everything,
cells.
you mean everything phagocytic,
Everything. Yes.
like they knock out dendritic cells, right?
Yep. They can knock out
messing with
specific
neutrophils.
kinds of B cells that are phagocytic. They can,
Yeah.
like, they are not a precise tool. This has been a problem in macrophage biology investigation since I was a grad student. This tool gets used as a very specific way to ablate things, and it is never a specific way to ablate things. So the big quibble that I have at the end is not that I don't find this plausible. It's not that I don't find it cool. I think it's probably happening. I would like to see an actual specific ablation of the cells that they are talking about and an inability for these pigeons to home, right? Because for me, the connection between these cells specifically and this homing phenotype that we're seeing hasn't been made yet. And I think probably it's doable, right? I think probably these cells, I mean, they pulled out, 7,000 or whatever specific genes. I'm sure that you could target a knockout to those at least temporarily, and do the experiment. So that's the only thing that I would say. It's one of those things that, like, as a reviewer, I think I would probably demand knowing full well what the answer is going to be, but just for the sake of continuity of the data, I think I want one more experiment.
So I don't disagree. So the thing that I'm kind of wondering is, like, do we think that is necessary for this science paper? Like, should this paper not have been published in Science, given that caveat?
Yes. And here's why.
You tell me why.
Because collagenate is inflammatory and hits every organ system. And you don't necessarily know here if an inflammatory response in and around the brain has a secondary effect that prevents these pigeons from homing outside of the direction of the sun. Like, it doesn't have to be an iron-loaded thing. This could be a phenomenon unrelated to magnetism, but related to inflammation that has this weird effect that, like, the animals feel crappy, and without the sun, they're not able to orient in the same way, right?
Yeah,
and I'm not saying that that's probable. I'm saying it's a possibility, and that's why I would have held it up.
If it weren't for the sun navigation, I would agree with you. But I think, like, the idea of a non-specific brain effect that is only related to whatever is the non-sun, I mean, totally plausible. It would have been cool if there was some kind of control where they, like, I don't know, give these this is not the thing they should have done, but just bear with me for a sec. If they had, for example, given these pigeons bird flu, I'm totally kidding. Not what you should do, but yeah, I guess given that, as I said, in this Ed Yong book that was just published last year, he's talking about how, like, these conversations about what is the sense organ have been going on for decades.
Yeah.
And no one has been able to plausibly show an organ system that might be involved. I think there was even a line in there, like, the person who figures this out is gonna, like, win scientific fame or, like, all that. I think that was in the book, if I recall correctly. So given how old this field is and how contentious this bit is, I feel like this is, in decades, seems to me, again, like, not being an expert in this field, this is the first, like, really plausible identification, potentially. I agree with you, there's a lot of caveats,
No, there aren't that many actually. There's just the one. I would
we
say
don't know. Like
yeah.
you were saying, like can we do a CRISPR based knockout? And what it sounds like you want though is you want like tamoxifen inducible like diphtheria toxin receptor expression. Like there's a bunch of genetic tools, a bunch of things that to knock out a specific cell type doesn't only require knocking out a specific gene. It might require all kinds of expression systems. Like, I don't know what pigeon promoters are. Probably somebody does. But, like,
Sure,
you'd
somebody
need to
does.
get the right gene under the right promoter. You'd need to, like, know does, if you're going to do DT receptor, like, do pigeons not normally have DT receptors? We know mice don't, but, like, there's a lot of genetic tools that you would have to, like, an entire paper, I think, just establishing a genetic system in pigeons if it doesn't exist. Like that's not a trivial thing to ask
trivial. It's not trivial, but it is a groundbreaking paper in science that to this point has some histology, some flow, some max, and then trained pigeons flying around. Now, I'm not saying that is not a lot of work. I'm certain that it was. What I am saying is that it is going to be the most cited paper in magnetic homing navigation for the liver. And I think that because of that one additional push for me would have been necessary. And this is why, by the way, it's completely reasonable for reviewers to come together on grants when they know what they're talking about and have discussions about like what is and is not reasonable. It's why we all don't come to the same conclusions all the time, but it's also why we need to know what we're talking about before we start giving people money. So, we're taking it away. Regardless, I think it was a great paper. I fully expect, regardless of whether they do that follow-up experiment, I fully expect that this will actually be the mechanism. It'll be interesting to see the biology of how this actually works. Be interesting to see, and it's so hard to study this. The idea of the liver being able to point the brain in a direction is a fascinating sort of consciousness question that I have. Like, what would that be like? To just kind of inherently know.
This is a plug. I
Right.
think that feeling that you have right now is a plug to read this book. Like, there's so many things like that where it's like, what would that be like to be able to have that? Like he talks about echolocation, like what is it like for a bat to be able to like really remarkable, like change direction like multiple times a second based on echolocating sounds that are like with lots of other bats flying around. So it's not, they're like getting their specific frequency of pitch and duration, but all the other bats of the same species are also bouncing. Like how do all that kind of stuff I think is super fascinating. It's very cool. I wrote this in a Blue Sky post, and I tagged Ed Young with a provocation on this paper saying, here's my hot take, is that the entire immune system is a sense organ. Discuss.
Interesting. Yeah.
The reason I say that is like, I think, for us, we are so used to thinking about senses in the way that they interact with our conscious experience. But clearly we are doing a lot of sensing that we are not conscious of, right? Like even for our most dominant sense, which is vision for humans, at least for sighted people, there's a lot that we see and even can react to that we are not conscious of seeing, right? Like things out of peripheral vision. There are things in our blind spots, there are optic blind spots, like all of this stuff and so I wonder the extent to which, and this is, just me doing the boundary fuzzing, that I'm interested in doing, but like, yeah, a big component of the immune system, certainly in the gut, is identifying and responding to signals about what we have eaten, what our nutritional state is, what our inflammatory state is. Lots of infections come that way. And so I do think there's a sense in which the immune system is modifying behavior based on sensing the world. And,
Yeah, I like that take. Kind of. I like the discussion around it.
Yeah, deliberately provocative. I think.
Yeah. Well, I think we've talked so much about the gut immune interface, right? As exactly that, as a sensing component of the entire system, understanding its health and boundaries with the outside world and all of that. I think it's probably. I think I immediately go to a yes and sort of place.
Sure. Which I think is totally appropriate. But yeah, I think this is a cool paper. An Immense World by Ed Yong. Definitely plug if you are interested in any of these topics. And yeah, I don't know, like, even if this is a macrophage, like what that has to do with what we normally think of as the immune system. But again, immune system showing up in weird places, doing weird things.
Yeah.
Theme of Autoimmunity Season 2.
That's right.
And I think we can leave it there.
Unless
you have anything else that you wanted to
No, I got myself in enough trouble last time.
Fair enough. All right. So this has been Autoimmunity, episode 57. I have finally updated the website. So autoimmunity.org now points to the Substack. AUDIOMMUNITY.org is our home base once again. Please like the episode and subscribe on whatever podcatching services you use. If you want to support us, you can subscribe at on our Substack page. Again, autoimmunity.org. Substack won't let us do less than $5 a month as sort of the baseline. But as I've said before, send us an email, contact@autoimmunity.org. We will give you a discount code for whatever you want to pay if you want to support us. Share us with friends if you think this discussion was interesting. We do still highly encourage you. There is still time as of posting of this episode for you to go fill out a comment on that OMB thing. We've got links in the previous episode 56, lots of links, including links to a AI tool put out by FASEB that will like let you put in your specific research, your specific things that you are concerned about, and it will sort of craft a original comment for you. You should edit it heavily. It's not like it's obviously AI written, so you should go in and edit it. But it gives you at least a framework. If you're interested, you can also just read the many posts written by scientists. This is one of those things, I think it has still yet to break out of the scientific world. I mean, more and more politicians are talking about it, but I think we need more people talking about it that are not scientists and contacting their representatives, making comments. So share it with your friends. Music at the beginning and the end is composed by Rachel Reinick, and we'll talk to you next time.
Carry on.
Sometimes you get lucky.